and the proteins that will help transfer the donor DNA to the recipient. Here's the origin of transfer, where a copy of this plasmid would be passed to the recipient. So these are nightmarish. There is a really quick. Okay, so perhaps some of you saw the frontline video for the nightmare bacteria. I'm hoping we'll have time so I can show you at least part of it, because boy, is it a wake-up call regarding antibiotic resistance. So perhaps in the popular press you've been hearing about these superbugs. These are bacteria to which we no longer have antibiotics that can kill them. And one example are the gram-negative Enterobacteriaceae that carry an antibiotic resistance gene that's called the New Delhi. Oh, I can't spell New Delhi. D-E-L-H-I, New Delhi metallo-beta-lactamase. And I know this is a long name, you guys, but there are many different types of beta-lactam antibiotics, different families of them. Some are resistant to, like the beta-lactamase that Staph aureus would make, right? So some of these classes of antibiotics, they remain like our last hope to treat some of these gram-negative bacteria. So now, the gram-negative bacteria have evolved a beta-lactamase that can destroy all of our beta-lactams, all of them, right? And these bacteria, they carry other antibiotic resistance genes, right? And often these are carried on a conjugative R plasmid. So the conjugative R plasmid, it carries the gene for this New Delhi metallo-beta-lactamase. It carries antibiotic resistance genes to a whole broad range of antibiotics. And the horror is that in India they've discovered that these bacteria that carry this conjugative R plasmid, they can conjugate with recipients in mud puddles in the environment. Now, before we always thought it had to be like a pretty specific environment, maybe like inside our intestine, right? So this is absolutely horrifying that these bacteria can conjugate and pass these multiple antibiotic resistant plasmids to one another out in nature in a mud puddle, right? So this program, you guys, it's excellent. And like I said, I'm hoping at the end of the semester we can watch at least some of it, because it is really does a much better job than I can ever do in describing the challenges that we're facing nowadays. Okay, the last topic we're going to do is on transposons. And transposons also contribute to the spread of antibiotic resistance. Transposons have been nicknamed jumping genes because they are DNA sequences which can move or jump from one place on a chromosome to another place on a chromosome. They can move from a chromosome to a plasmid. They can move from a plasmid to a chromosome. So hence jumping genes. They were first described in the early 1900s by a plant geneticist back at Cornell called Barbara McClintock. She was studying the beautiful native corn, the beautiful corn that has all the multicolored kernels, right? So she was trying to understand the genetics of pigment production in the Native American corn. So she lived in her cornfields. She had her little shed still there. You can visit it. She lived in her cornfields. She knew individual stalks, right? And she knew generations. She knew, like, oh, this is the grandchild of this plant. So she just knew the corn intimately. So she was the one that came up with the educated guess that color production had to depend on these jumping genes. Like a transposon would jump into one pigment gene and knock it out, so that color wasn't produced. Then it would potentially cut itself out and the color would come back. Or possibly a transposon would enter a site that actually turned on pigment production. Right, so they all told her she was crazy. Barbara, you've been spending too much time in the cornfields. Right, that she was pretty tough. She kind of stuck when other geneticists started exploring, this is a model, it turned out she was right. So transposons we find not only in eukaryotes, right, plants, they're rich in bacteria. And as we might guess, of huge concern is the transposons. This is called transposon 5. This is a gram-negative transposon. What can they carry? Antibiotic resistance genes. So one model for evolution of the plasmids that carry multiple antibiotic resistance genes is that the plasmids slowly accumulated the antibiotic resistance genes with the help of transposons. Right? So different transposons carrying different antibiotic resistance genes would insert into the same plasmid, and over time the plasmid started accumulating a variety of different antibiotic resistance genes. So again, kind of bad news for us. Okay, so folks, there are additional slides that have to do with inappropriate or overuse of antibiotics and how that creates an environment through natural selection. We select for antibiotic-resistant bacteria. Please take a look at those slides because it's an important topic. And there's also some genetics vocabulary that one of your colleagues said was on the TEASE test. So like phenotype and genotype, those are two genetic terms that phenotype is how the organism appears, where the phenotype is products of the genetic information. Genotype is the genetic information of the organism. Right, so those are two terms for the TEASE test, and you need it. Those are two terms to have at your available. Okay, guys, so